Okay. Welcome everyone. It is my pleasure to introduce my friend and tonight's speaker, Yiva Lekberg. Yiva and I met at the River Road Farm in Missoula in 2020. Actually, I think I was the caretaker at the farm at the time, and I looked out of my window and it was like snowing and cold, and my dog was like barking at the window, and this figure was out in the field like plucking, like overwintered spinach. And I then like looked at my phone, and my farmer boss was like, "Oh, by the way, my friend Ilva is gonna be coming by to grab some greens." And so this lady was out there foraging greens in like February in the snow. Um, but then we actually met. She was coming on a weekly basis to volunteer at the farm. And we just immediately hit it off talking about fermentation of food. We were talking about kimchi and sauerkraut. And I think we went on a deep dive into kombucha and sourdough. We might have even exchanged some sourdough starter. I don't know, but it was one of those moments where you're like, oh yeah, I love talking to this person. So and if you're out working in the field, a good conversationalist is golden. But I also know that Ilva is not only interested in the edible microbiology, but also she is a real genius in the world of ecology. She is a soil ecologist at MPG Ranch and an adjunct professor at the University of Montana. And her research focuses on structural and functional shifts in soil ecosystems associated with plant invasions and how these changes affect restoration success. Prior to her work in invasion biology, Ilva explored the role of mycorrhizal fungi in subsistence farmers' fields in sub-Saharan Africa, coastal grasslands in Denmark, and geothermal areas in Yellowstone National Park. So I'm so thrilled to have her to top off our amazing list of programs this season. I did ask if she was going to give us some hopeful news, and she gave me a very non-committal answer. So I can't wait to hear what she has in store. Welcome, Ilva. Thank you so much for joining us tonight.
Thank you so much, Alex. Can I start sharing? You got
Yes, here,
it.
let
Stop
me get out, let
sharing.
me stop sharing. Let's see. Oh, stop share. Yes,
Let's
go for it.
see if I can get this to work. Thank you, everybody, for coming. Hello, let's see. Now I'm digging myself. Okay, there we go. Can everybody see the full screen? Okay, great.
Great.
Thank you so much, Alex, for that nice introduction. Yeah, River Road is kind of my happy place when I need to just get away from the office a little bit. Going and weeding and having somebody tell me what to do is lovely. And then having great conversation around food and fermentation has made it even better. So I don't know how many of you guys are from this area, but if you are from Missoula, you might have seen this photo before. It's from Mount Sentinel, right on the backdrop of University of Montana. And this photo was taken sometime in the 60s, early 70s, and then 40 years later. So you see this like beautiful mixed native grassland has just been turned over to a pretty much monoculture of spotted knapweed. Oh, I was worried that this was going to happen. Oh, there we go. There we go. It was a little slow. So I work with MPG Ranch. And again, like for those of you who are not in Missoula, and even if you are in Missoula, you might never have heard of us. We are a privately funded research organization that is located in the Bitterroot Valley on a 17,000 acre property. It's a former cattle ranch, and as such, it is heavily invaded by weeds that we research. We also do research related to restoration ecology, and then, as you know, what I do, invasion biology, and then plant herbivore interactions. We, in addition to that, we have a pretty big educational outreach and have about 2,000 visitors a year. And if you are curious, we also have a website. So before I start sharing what we've learned since I started in 2010, I want to acknowledge some of my coworkers that have been instrumental in gathering the data and analyzing the data that I'll present today. So out on the ranch, we have mostly 4 exotic plants: cheatgrass, leafy spurge, knapweed, and sulfur cinquefoil. And I'm sure you're familiar with them. Somebody has a radio on or something.
See, if everyone can just make sure they're muted.
Okay. Yeah, it's just a little distracting. Thank you. Yeah, so the main weeds that we have on the ranch, and I'm going to use weeds and non-native plants, exotics interchangeably. With an invasive plant, what we think of is one that is non-native and then is just replacing native plant communities. So these are the 4 main invasive plants that I'm going to talk about today that are really prevalent in our area. Oh, it's just not working, the forward. There we go. So one thing I was really fascinated by when I started thinking about plant invasion in 2010 was how these exotic plants can come from Europe or Asia and then outperform our native plants that are locally adapted. So they come from a completely different environment, and in many cases they can double productivity, even though they're exposed to exactly the same conditions in terms of nutrients, water, temperature, and light as the native plants. So how do they do that? And that is something that has occupied a lot of my thought for the last 15 years. So it's true for many invasives, invasives in general, but it's also true for the invaders that we have in our area. So this is data collected by Morgan McLeod for her master's thesis. And what she did was simply going out to Mount Sentinel, Mount Jumbo, the North Hills, and some other places, and found invaded patches and also adjacent native patches. And then she just measured above ground biomass. So basically what you're seeing here in this graph is above ground biomass clipped on a little square. And all the invaded patches are in black. And you can see for cheatgrass here, knapweed, leafy spurge, and cinquefoil. And then the adjacent native patches are in white. And you can see for the 3 out of the 4 invaders that, the productivity is so much higher for the invasive plants. And you've probably seen that too when you're out hiking, right? You come across a spurge patch or a knapweed patch, and you just see that productivity is much higher. Of course, when you go out to the native area, you don't know if the invader created those conditions for the increased productivity, or if they just invaded areas that were already disturbed and therefore have higher nutrient availabilities. So in order to get around that and try to assess causation, in 2011, I was able to convince the ranch crew to actually plant a lot of these invaders. So we set up this common garden experiment on the ranch where we had monocultures of knapweed and cheatgrass, sulfur cinquefoil, mixed native communities of representative grasses and forbs, and then leafy spurge. So we had 5 reps of each plant community type. And then it's just going all over the place. And then we've just followed those communities over time to see how does productivity change, how does microbial communities change, and nutrient availabilities. And the beauty with having an experiment like this is that the conditions were the same starting for all of the invaders. And if we're seeing differences over time, we then know that it was created by the invader. The other thing we were interested in is that these invasive plants are very different from each other. For example, you have leafy spurge, is a perennial deep-rooted forb. And then you have cheatgrass, which is a shallow-rooted annual grass. So you can imagine in terms of effects on below-ground microbial communities, they can be very, very different. So we were interested in, do invasive plants always do the same thing, for example, increase productivity, but do they have different ways of doing it? And after a couple of years, we did see that typical pattern in the experimental plots as well. And this is just plotting biomass of the native mixed communities, cinquefoil, cheatgrass, knapweed, and spurge. Again, from a small area within each plot. And as you can see here, we see the same thing as we see out in the field, that biomass production was lowest in the native plants, but then higher in cheatgrass, knapweed, and spurge. And cinquefoil being somewhere in between, and we see that quite a lot. It doesn't seem like it's a super aggressive invader, and productivity is not always higher than the native communities. So related to, we know that plants need nutrients, right? The nutrient they need most of is nitrogen. So it makes sense when you're thinking about invasion and increased productivity to then also think about nitrogen. And Morgan did that as well. So this is the same kind of setup as I showed you before with productivity, but now she measured nitrogen availability in the soil. And as you can see here again, 3 out of the 4, cheatgrass, knapweed, and spurge had much higher, like in some cases more than double nitrogen availability compared to the adjacent matched native patches. And again, like sulfur cinquefoil didn't really show any difference. And this seems true in general because this study by Liao et al. Summarizing 94 papers on invasion, plant invasion, showed that invasive plants contain on average 85% more nitrogen than native plants. And that because they have more nitrogen in the shoots, litter decomposition is also ramped up. And that often results in increased nitrogen availability in the soil by up to 50%. So again, when we're thinking about nitrogen, we should think about microbes because they are in fact involved in all of these transformations of nitrogen. And just taking a step back, that soil is not just dirt, right? It's a living, thriving ecosystem below ground. And I'm sure you've heard this before, but it's worth repeating. Like 1 teaspoon of soil can contain up to 1 billion bacteria and several yards of fungal hyphae and thousands of these like protozoa, and then 100 to up to 100 nematodes that then eat both these microbes as well as sometimes plant roots. This I'm just putting up here to remind you of the nitrogen cycle. You might have had to study that some point in school, but you know, we don't have to go into in detail. All I want to do here is just to highlight everything is red here is transformations that are driven either by fungi or bacteria in soil. So you can see anything from nitrogen fixation, right, to transformation of different nitrogen species in the soil up to where nitrogen is also lost from the system. So it makes a lot of sense with invasion because of the higher productivity, higher nitrogen, to also look at soil microbes. So, and that's what Morgan did in her study. Again, this is the same thing. She sees higher productivity, higher nitrogen availability. And she then focused on the abundance of these ammonia oxidizing bacteria. And they're the ones that convert ammonium to nitrate. And nitrate is a source of nitrogen that most plants and grasslands prefer. And what she saw here was that all of the 4 invaders had higher abundance of these ammonia oxidizing bacteria than the native plants did. So they somehow changed the relative abundance of this bacterial group that converts ammonium to nitrate and make nitrate more available for then uptake and converting to biomass. And again, like relating to this analysis across invasive plants, it seems like microbial biomass often increase with plant invasion. We look at microbes, of course, they are microbes, so it's hard to you can't just take up some soil and see that there are bacteria there. We actually have to use some molecular techniques to characterize microbial communities. You might be familiar with it, but in case you're not, I just wanted to very briefly go over how we go about that. So we always start with either soil or plant roots, depending on the organism we're looking at. We take that back into the lab, and then it's basically just baking. You just follow a protocol, and you start with soil, and then in the end, you just have pure DNA. And that DNA we take then and amplify, multiply many times through a process that we call PCR. And that allows us to amplify it so much so we then can send it off for sequencing. And then we get those sequences back, millions and millions of sequences. Then it's like complicated bioinformatics that I am not good at because I'm really not good with computers, but fortunately my colleagues are. So they can sort out all these sequences into what samples they belong to. And then because we have the sequences, we can use databases online to get a match and identification. So we can say like, okay, this bacterium is this taxon, this is this taxon. And with fungi, we can get even further using another database and saying, is it a potential saprotroph? Is it a potential pathogen, or is it a mycorrhizal fungus? So with fungi, we can get a little bit further and looking at potential function. So with all that information, we get like a lot of sequences related to each plant species or soil sample. And it's obviously a lot of data. One way to look at that data is to put all that information into two-dimensional space. And I'm going to take some time and walk you through this. This is what we call an ordination. And all you need to know really is all that microbial community. In this case, it's bacterial communities. And so here we're seeing like we take all that information, put it down in two-dimensional space, and the closer 2 points are together. Here, for example, you can see that knapweed and native mix in our experimental plots are very close to each other. That means they actually harbored very similar bacterial communities compared to, for example, spurge and cheatgrass that are really far apart. And for spurge and cheatgrass, I'd be like, yeah, that's what I would expect. They're very, very different plants. So it makes sense that they structure very, very different bacterial communities. But what this is telling us is also like when these plots have been growing, I think this was like 4 or 5 years after we planted these, and the bacterial communities did not differ when we started the experiment, right? But over these like 5 years, these plants have structured unique bacterial communities that are different from native communities. And except for knapweed, for some reason, they're very similar. And we see that across fungi, bacteria, and arbuscular mycorrhizal fungi, which is very curious to me because knapweed is so different from native plants. And I actually have no good explanation for that. But we'd also see that the other plants structured unique bacterial communities. So in this case, even though they all increase productivity, the bacterial communities they structure are different and species specific. In these plots, we were also interested because we're seeing differences in nutrient availabilities. We also see differences in bacterial and fungal communities. We were interested to see what the effect were on plants. So what we did was what we call like a full reciprocal transplant. And this was a study we did together with a high school student, Leela Marsh, that came out in one of those Bitterroot wildlife internships that we run every summer. So basically, within each plot, you can see it down here, we planted seedlings of cheatgrass, knapweed, spurge, sulfur cinquefoil, and then either blanket flower and blue bunch, so two representative of native plants. And then we grew them for 2 months and we watered them as needed. And all we wanted to do, wanted to see was like, do invasive plants grow better in their home community? Or are they somehow structuring the soil to favor their own growth? Or are they favoring other species? For today's talk, though, I'm going to focus on the two natives primarily. So this is because this is a native plant species talk, not really native plant species talk, but native plant society. So we had blanket flower and blue bunch. And this is blanket flower planted into either native, cinquefoil, cheatgrass, knapweed, or spurge plots. And the same thing with blue bunch. And this is just the biomass of individual plants. So you can see here that biomass varied among plot types. So it mattered what plots blanket flower and blue bunch were planted into. What you can see here also is that both of them really loved spurge. And I think the main reason there is that spurge has so much higher nitrogen, and the biomass of these plants were enormous when they were planted in spurge. Again, here they weren't competing with spurge because we kept the adult spurge plants away. So there was no really plant-to-plant competition that you would see out in natural invasions, but it just shows that the soil has changed and that has consequences for plants. But here I'm just throwing in the contrast of cheatgrass, right? Like, so it's the same with cheatgrass. You can see that it loves spurge too, but look at the y-axis here. Here we get up to 800 milligrams, and here we're at 5,000 milligrams. And that again just highlight that, you know, invaders are very good at taking available nutrients and turning it into biomass. And I don't know if you, when you're out hiking, if you ever see this, but now when I know this, that cheatgrass, it loves nitrogen so much, you always see it together with leafy spurge. And so next time you're out hiking, look at this, the cheatgrass in a spurge patch compared to when you see cheatgrass in native communities, and they're so much smaller. And that is because I would think that it's because of the higher nitrogen availabilities in the spurge patches. So just a first kind of wrap up. We know that invaders can often increase productivity and increasing nitrogen availability. And we often also see shifts in bacterial communities. And that is likely what drives that higher nitrogen availabilities. For restoration practitioners that want to try to restore these invaded communities back to native grasslands, know that nutrient availabilities are often higher. So you have to deal with that somehow. And there have been different thoughts of how to do that in terms of maybe adding sawdust or sugar. I don't know, they mix successes, and sugar is obviously expensive and it depends on the size you're trying to restore. Just keep that in mind, that nitrogen availability is probably higher. And, you know, because of that, it also makes you really susceptible to invasion meltdown. Like we saw with cheatgrass, another invader is coming in and just like really taking up that space. One thing that we don't know much about is how long these legacies persist. Is it a few months? Is it years? Does it depend on if we're looking at nutrient availabilities or microbial communities, for example? We've done a little bit of work on that in these experimental plots that I'm going to share with you later. But before I do, I cannot give a talk without talking about my favorite microbe, and that is mycorrhizal fungi. I'm a mycorrhizal ecologist at heart. That's my comfort zone. And I'm sure most of you have heard about mycorrhizal fungi, but if you haven't, it's this symbiosis between plants and fungi, where the main function is that fungi provide nutrients in exchange for carbon. And I think there's no better picture that illustrates this than this small little pine seedling. So you see these roots just extend very short here, and all this white stuff is fungal mycelium, right? So that obviously gives this plant access to so much greater soil volume to explore for nutrients. And you can also imagine that it's quite costly for the plant because all the carbon required to build this huge mycelium comes from photosynthesis of this little shoot. So the plant better get something good for it, you would think, if you're spending that amount of money, or carbon rather. What I'm going to talk about today is arbuscular mycorrhizal fungi. And they are formed between mostly herbaceous plants, so it's relevant in grasslands because they form this arbuscular mycorrhizal mycorrhizae, not ectomycorrhizae. They colonize 90% of all plant families, and because they're so common, it's resulted in these sayings that plants don't have roots, they have mycorrhizaes. I already told you what they do, but because they're so abundant and they occur in this root soil interface, they could be very important for invasion, understanding invasions. And what we're looking at here is just basically roots growing in an agar plate, and then you have hyphae growing out and then spores forming here. And what we're seeing here on the right is a staining technique that allows us to go and look inside the roots, because these fungi are not just only on the outside, they also penetrate the root and occur inside the root. Of course, roots are not blue. We just use a blue stain because the fungi absorb more of that blue stain than the root. And it allows us to see, like, this is the outside of the root right here. And all these dark blue structures are arbuscular mycorrhizal fungi inside the root. These are these vesicles, or maybe spores. Sometimes they look very similar. They're basically just lipid bodies that the fungus store carbon that it receives from plants. And then you have these like tree-shaped forms. That's why it's called arbuscular. It's arbuscule, tree shape. And that's just this incredibly fine tree-like structure that where the exchange of nutrients and carbon is occurring. So one common feature with mycorrhizal fungi is that it can increase biomass a lot. This is pigeon pea that I grew for my PhD in Africa, and I had grown them in sterile soil without mycorrhizal fungi and with mycorrhizal fungi. And, you know, obviously this plant is having access to, oh, it does jump around a little bit. This plant has access to the same amount of water and nutrient as the plant to the left. It's just that the fungus is there in the pots helping it take up nutrients. It's not always beneficial, but if phosphorus is limiting especially, it can be beneficial. It can also help with drought tolerance and pathogen protection, again, in exchange for carbon. Related to invasion, though, there are two main hypotheses that invokes mycorrhizal fungi. One is the one we call enhanced mutualism hypothesis. It was coined by the professor here at Ray Callaway. And that posits that invasive plants have a higher mycorrhizal dependency and benefit more from mycorrhizal fungi than native plants. So they come in, they increase that benefit, and then they allow them to outcompete the native plants. And there's some support, as I said, for knapweed supporting this hypothesis. As in everything else in ecology, there is a hypothesis to state the complete opposite. And that is the degraded mutualism hypothesis, arguing that invasive plants are predominantly non-mycorrhizal or have a low mycorrhizal dependency. And garlic mustard, I don't know if anyone here is from the Midwest or New England, it invades a lot of these understory deciduous forest, can make a delicious pesto of it, but it is non-mycorrhizal. So when it invades and increase in abundance, it will basically starve the fungi. So they don't get any carbon from it because they don't form that association fungal abundance will decrease, and that reduces then the competitive ability by native plants. So we were curious when I first came and started on MPG Ranch to assess how invasive plants change mycorrhizal fungal communities and abundance. And we wanted to expand beyond the ranch and have like replicated sites to see if we saw changes, if they were consistent between sites. And again, if different invaders did different things. So we found if you're from the Missoula area, you know these places, and each location, we found these 4 different plant communities. This was before I actually had discovered sulfur cinquefoil, because he was kind of hiding out there on the ranch. So this only focused on the 3 that are in your face, like spurge, cheatgrass, and knapweed. And we paired that also again with mixed native communities that sometimes were great and sometimes somewhat degraded because they had been sprayed. Like a lot of these native communities have been sprayed by Tordon, for example, that kills all the forbs. So they're often grass dominated. But we sampled roots from all of them, and then we did the molecular analysis that I said, then characterized the AMF communities. And then we also took root segments and looked to see what proportion of root segments had mycorrhizal fungi. So in some cases, I'm going to show you percent colonization. And that's just the proportion of root segments we looked at that were colonized by mycorrhizal fungi. So this is again like the first looking at just the abundance. So again, percent colonization in the remnant native communities we had in these 6 locations: the cheatgrass, knapweed, and spurge. And again, like if for 80% colonization, 80 out of 100 then were colonized by mycorrhizal fungi. So you can see here, like if 40% is roughly what the native communities have, when cheatgrass comes in, it suppresses mycorrhizal fungi. It behaves similarly to like garlic mustard. Cheatgrass is not a non-mycorrhizal plant, but it has if you think about it, it's a fast-growing, shallow-rooted annual plant. All they want to do is produce seeds. They don't want to invest in a symbiosis below ground and give him then carbon that might then compete for seed production. On the other hand, when you have relatively coarse-rooted perennial forbs, they really benefit from mycorrhizal fungi, especially spurge. I've grown that in the greenhouse and it barely grows without mycorrhizal fungi. So they have a vested interest in investing in this symbiosis because they're perennials, right? So they are more supportive of this enhanced mutualism hypothesis. So we have both, and it seems like it depends on the invader here. So when we were thinking about the ammonia oxidizing bacteria, then all the invaders did the same thing. And in this case, with mycorrhizal fungi, it kind of depends on the plant functional group. And that's what I've spent a lot of time trying to understand, like when do invasive plants do the same thing, and when provenance matters, and when do they more behave like a typical forb or a typical grass. And I still I haven't figured that out yet. You know, I think with mycorrhizal fungi, I can pretty much predict what's going to happen, but with many others, I can't. So we were surprised in this. We also looked at the richness of fungi and diversity. And here we thought, okay, we have a mixed native community that is being replaced by a monoculture weed. And the thinking at the time was that there is a connection between above ground and below ground diversity, because different plants favor different fungal species. And the more plant species you have, the more fungal species you would have. In this case, though, that's not what we found. If we're just looking at the AMF richness here, it's about the same for the native mixed communities and cheatgrass. But then look at knapweed and spurge. They are so much higher than the cheatgrass and native. Same goes for diversity when we're taking relative abundance into account. So you go out to a monoculture, and this was true across the 6 locations that we looked at. So it's not just one. It seems like it's a pretty general phenomenon. And to me, that also indicates that, at least for mycorrhizal fungi, what matters more is probably carbon allocation, the amount of carbon that is allocated below ground, rather than the number of species that you have in the plant community. Again, if we go back to, I know I'm showing a lot of data, but just bear with me here. If we go back to the experimental plots, we might ask also here, do they structure or vascular mycorrhizal fungi in species-specific ways? And you can see that they do, right? So again, like here's knapweed, here's native, and they're closer together, not as close as the bacterial communities that we saw, but they're still the closest together. Whereas like leafy spurge and cheatgrass are again the furthest apart. So here we can say that yes, the invasive plants, it wasn't just that, I mean, we saw it in 6 different locations and it was pretty consistent there. But even on this small spatial scale, they sort themselves, the AMF communities sort themselves according to the plant community. We were wondering then, we knew that like the AMF abundance and the community differs. Will that then translate into a native plant if we plant that into all these communities? So going back to that study we did with where I showed the big cheatgrass, we then looked at the colonization of the blanket flower roots. And we saw here, so this is again like root colonization, 30 out of 100 intercepts were colonized by mycorrhizal fungi if we planted them in the native plots. And that was suppressed in the cheatgrass plots and then trending higher in the knapweed and spurge. So this is kind of the same as we saw in terms of the abundance in the 6 field locations. We're seeing the same thing in the experimental plots. So we can probably say, be fairly certain that when cheatgrass invades, it's going to suppress AMF abundance and diversity. And when knapweed and spurge invades, it's going to likely promote it relative to remnant native communities, especially if those remnant native communities are dominated by these C3 grasses that we have, like blue bunch wheatgrass and June grass, because grasses in general, unless they're C4 grasses, are generally poorer hosts to AMF, meaning they allocate less carbon. This is a lot of text, but basically it's just what I said. We see shifts in AMF communities. They are invader specific. And that we can have some predictions based on the mycorrhizal association by the invader relative to the native plant community that is being invaded. So if an invader is a mycorrhizal forb, AMF abundance might be higher. And the opposite being true if it's an annual or short-lived grass. So I would say like there is this sometimes a focus on the need to inoculate as part of the restoration practice. There are a lot of companies now that sell inoculum. I work with farmers too. They're also inundated with these products. Some of them are good. Many of them are not. They've even done some screening and showing that a lot of them actually contain pathogens as well. But more importantly, you spend a lot of money to bring in a fungus that may not occur naturally in your soil. So if you want to try to restore a former cheatgrass plot, for example, or if you even know that the weed is non-mycorrhizal, there are always some propagules there. So try to promote what you have there instead of bringing in something else. So you can do that maybe by growing one of those like bridge species to promote the soil conditions and microbial biomass before you then start restoration. If anyone is interested in that, we can talk about that after the talk. There is plenty of time, I think. Again, similar for the bacteria, we don't know how long those legacies persist. So the last thing I want to talk about is work we've done, again, in these experimental plots. So we know that biomass differed, bacterial communities differed, and fungal communities differed. then the next logical thing is to ask, what are the consequences of this for restoration? So in the fall of 2021, we removed all the above ground biomass when the plants had gone dormant. And then in the fall, we sprayed it a few times with glyphosate. We didn't want to till or anything to disturb the soil, but we needed to kill the weeds, anything that grew back in the fall. So we used glyphosate a couple of times to try to flush out some of the seed bank. And then in the spring, we divided all the plots into 4 1-meter squares, and we had 4 treatments because we were interested in seeding versus transplanting. We know that most restorations, they go out and seed because often that's the only thing you can do, but they often fail. And in this case, I was also interested in looking at legacies of these invaders. So I needed some plants to survive. So in this case, we also transplanted seedlings into it. So we had one treatment where we seeded and either weeded or not, we just left it, or we transplanted seedlings and weeded or not. And the forbs that we chose were forbs that are, common in our area. I don't know with flax how common it is, but we chose it anyway. But all the other ones occur. And here are the 4 grasses that we had. Yeah, not much more to say about them other than we have them on the ranch, and yeah, we wanted to try them. So this is what they would actually look like. This is one that was transplanted and weeded. You can see that it's pretty good. Like it has big penstemon. You see some of the asters and blanket flower. And then you see the ones seeded, not weeded, or transplanted, not weeded, completely a weed fest, right? And I'll show you some data on that. What struck us was that a lot of them, and maybe it's not too surprising because we're surrounded by ag lands, but a lot of them were ag weeds. So we see a lot of these mustards. The other thing you can see here is that the seeded, weeded one, this was actually a good one. In some plots, we didn't have anything come up from the seeding. So today, I'm actually not going to talk about the seeded ones because it was a dismal, it was just a dismal failure, to be honest. But I think it also reflects what happens in a lot of these restorations. You put a lot of seed out and it often doesn't work. So I'm going to talk about the transplanted, weeded or not weeded for the last couple of slides. This is just, again, it's a lot of data, so bear with me here. I'll walk you through it. Because we've been interested in nitrogen, and nitrogen is so important for biomass production. We measured 2 months after planting these seedlings in the different plots, soil available nitrogen in cheatgrass, cinquefoil, knapweed, spurge, and native. And first of all, like they're high. Like normally in a grassland, you might find something like 5. And here you get like almost 50, right? The other thing you're seeing is like, who's the king here? It's the spurge again. It's just like has so much nitrogen. In fact, it has so much nitrogen in its shoots that it rivals lupin, which is a nitrogen fixer. And it's fascinating because we have no idea where that nitrogen is coming from. We've tried to think about, because these ammonia oxidizing bacteria that I was talking about, they don't fix nitrogen from the air. They're just like converting ammonium to nitrate. But where is that ammonium coming from? And one idea is that they're actually breaking down organic matter because there's a lot of nitrogen in organic matter. But look what happens after a year after. Then we're back down to these more normal values again, either because the plants have taken it up or it's been leached from the system. So we don't know which one. But even these differences here early on, that's when you have seeds, that's when you have seedlings growing, and that could have a big effect on the trajectory of the community this early on. And especially if you're going out and spraying, you have this enormous nutrient flush. And if you have an invader there or an exotic plant, you're going to be very susceptible to this invasion meltdown. This again is one of those like, or two ordinations. I'll walk you through that too. Here's the bacterial communities 2 months after planting, and then again 14 months after planting. And you can see the bacterial communities, they show less, like, you know, they're more clustered here, but big year-to-year variation. If we're going to the fungi, on the other hand, you see the dispersal here 2 months after planting. We knew at the end of the experiment that we had these clear differences among plant communities. Well, 2 months after planting, we still see that. But then at 14 months after planting, they've kind of started to come together again, suggesting that, you know, the legacy of the fungal communities, we start losing it after like 14 months to 2 years. But again, like planting in those seedlings and those early differences could have an effect on the trajectory. And I'll show you that data, the last thing I do. But before I do, I would just want to show you here that in the non-weeded transplanted plots, most of them were weeds. And you saw that in the photo that I showed as well. So here, for example, these are these stacked bar plots. We'll go over the leafy spurge first. This is the legacy cover. This is the cover of leafy spurge in the leafy spurge plot. So it's less than 5%. And then the other ones are just other weeds, right? Like these mustards and whatnot. And here we have, you know, maybe 15% or so of native cover. So that was true actually for spurge, cinquefoil, knapweed, that the cover of those plants that came back was very small. And most of the weeds were other weeds. And that could partly be because we try to remove the seed bank, right, by watering and spraying. But it's still curious because these have been dumping seeds in there for 10 years. And yet when you try to restore it, they don't really grow back. So that was interesting. And except for cheatgrass, you know, that's the one, like half of the weed cover was actually the plant that used to grow there. This, I believe, is the last data slide. And that is showing the weeded plots that we have, transplanted weeded plots, and just thinking about what are the effect on just looking at the functional group, like grasses versus forbs with native plants, and does it matter what community they were planted into? And you can see that it does. So for example, here's the native communities. The forbs did really well here. So the ratio here is 3-to-1 based on cover of forbs to cover to grasses. Then you go to cinquefoil, they actually favored the grasses. So the grasses did really well, but the forbs did not. And there is this trend that if you were an invasive forb, you tended to favor grasses. And if you were a grass-dominated native or cheatgrass, forbs did better. So there's this like switch back and forth there. And that to me does not, you know, suggest the role of mycorrhizal fungi, that is that the community is affecting something in a beneficial way. It's more like we have pathogens here that are specific to functional groups. So in the forb dominated or the forb invaders, they favor pathogens that then transmit to native forbs. Whereas if you have grasses, they might have grass pathogens that then carry over. So possibly what we're seeing here is a signal of pathogen transfer between like the invader then to the native community. And we're currently looking into that to see if that is the case. So just to summarize everything, just some quick take-home messages. Microbial communities differ among invaders, and they seem to be invader specific and predictable and are caused by the invader. And we can say they're caused by the invader because we did a lot of that work in the experimental plots where conditions were the same when we started. I've also shown you that shifts in microbial communities coincide with changes in ecosystem processes, especially nitrogen availability, which is so important for biomass productivity. And then that these differences that we're finding in microbial communities and nitrogen availabilities can affect restoration outcomes. So I don't know how positive that is, Alex, for, the outcome, but that's kind of what I have. So I'm curious to take any questions that you might have and see if I can answer. Thank you for your attention.
Thank you, Ilva. So
I don't know
fast.
if I should stop sharing or we can keep it in case we need to go back to some slides.
Yeah. Yeah, you might end up switching back. We do have a number of questions in the chat, so I think I'm just gonna
Great.
I think I'm gonna go through those, and then I hope people will stick around and ask questions if they have more. I think the hand, if everyone knows how to use the raise hand tool, that might be helpful, but we'll start with these questions in the chat. Um, so Sharon asked, did you have a control for a native plot with no invasives?
In the experimental plots that we grew, yes. I am so sorry if that was not clear. Let me go back where we had the outline. Yes, we did. We had here we had 5 different community types. So there were 4 invasives that were monocultures, and then we had these communities. You can kind of see it there, here, and they were representative grasses and forbs, actually very similar to the ones we planted in the restoration after. So it was blue bunch, wheatgrass, we had blanket flower, but they were mixed though, so they're not monocultures.
Mm-hmm. And then Sharon also asked, I think, is it your hypothesis that invaders bring in their own AMFs?
Yeah, that's a really good question. And I get that a lot when I've shared this data because the richness is higher, right? So you would think that that's what they would do. But if I look at the community of the invade that increase, that explains that increased richness, there are these rare taxa that seem to have kind of been lost in the native communities. So I think more what happens is that the invaders come in, and because they allocate more carbon below ground, there are more taxa that can stay on. Competition can be tricky, right? Because if it just dumps carbon below ground, the most competitive one might outcompete the others. But I think in this case, the amount of carbon that goes below ground allow these rare taxa to remain. And I think the reason we've lost them in a lot of our grasslands is that we have sprayed them a lot. Like if I would go out to a really nice, pristine grassland that has a relatively high forb diversity, I would probably see a higher AMF richness. But if you think about like the weed management that has happened in a lot of our grasslands, where a lot of ranchers, they want grasses, they don't want forbs. So if knapweed, and also if knapweed and spurge comes in as forbs, you don't want to spray the grasses. You use a forb specific herbicide like Tordon. Like the ranch has been helicopter sprayed by Tordon twice. So a lot of them are just really grass dominated, and grasses are poor hosts. So I think that's what's going on, but it's not that's what I thought too first before I started digging in and looking at the community. But good question.
And Sharon gave a fascinating talk on trees earlier this season. So
Oh,
yeah,
nice.
thanks for coming, Sharon. Thanks for
I
participating.
saw Leela too. Leela gave a talk on mimulus. We worked
Yes,
together. We
yes.
worked together on mimulus in Yellowstone.
Oh,
Many
that
years
makes,
ago.
yeah, that makes a lot of sense.
Yeah.
You probably know many of the people who've spoken on
Yeah,
that.
maybe.
Beth, thank you for co-hosting. And Beth asked, please talk more, or said, please talk more about bridge species to promote natural AMF increases.
Yeah. Really, Chuck Casper is the restoration ecologist out on the ranch. I'm more the soil ecologist, but I can imagine sometimes it's hard, right, to go from your spraying a cheatgrass field and then just immediately planting native plants and expecting that to work. And especially forbs that might be more dependent on mycorrhizal fungi. So what we've been playing around with out on the ranch is cinquefoil, for example. It's a legume that you can grow. It establishes easily, quickly. It's a good host for mycorrhizal fungi. Many legumes are, because nitrogen fixation is a very phosphorus demanding process. So there's often associations both with nitrogen fixation, nitrogen fixing bacteria and mycorrhizal fungi. I've always wondered though with sandfine that because it's a nitrogen fixer, it might also increase nitrogen availability in the soil. And that's the last thing you want when you're trying to get rid of invaders, right? Because they're going to take that nitrogen and convert that into biomass. So I've had reservations against that bridge species, but the idea is very, it's pretty straightforward. You take one that can establish easily, that is relatively cheap and preferably short-lived, that you then terminate somehow. If it's short-lived, it's just going to die. But during the years where it's growing, or year where it's growing, if it associate with mycorrhizal fungi, it then provides that carbon, so you can start building the mycorrhizal fungi again. Of course, like it can't really build diversity if it's low. That can only increase by species dispersing in. So, but you're not going to combat that by inoculating, because you're usually just inoculating with one species. So thinking about it, I'm happy to talk to anyone. Like in the beginning of the presentation has my email. So if you have any questions or want to talk more about a particular topic, I'm happy to do that. So just reach out. But there are some traits with bridge species that you would be looking for, and it being mycorrhizal would be one of them, and short-lived. We've actually used yarrow out on the ranch. It's cheap, it's native. You can get a lot of seeds. And it wasn't meant to be a bridge species, but it did really well. It established everywhere so quickly. It was almost like a yarrow monoculture. And we were wondering, like, how are we going to get rid of yarrow now? Like, we don't want yarrow everywhere. But then it just died. Like, 5 years in, it was gone. And we were able to, because we had another experiment where we actually had some fungicide trials, just spraying some plots with fungicide, and it survived longer there than in the control plots. So that gave us an idea that, okay, maybe it's just pathogen buildup. Like that often happens. You have density dependent pathogen accumulation. We see that in agriculture all the time, right? That if you're growing a monoculture, you're going to favor certain pathogens. And we think that's what happened with yarrow, because we were able to correlate the decline with the increase of this pathogen. Now, is that a good bridge species? Maybe, maybe not. Like if that pathogen then is transmitted to other native forbs, it's not a good bridge species. So I think you just have to kind of think about and try it out to see first. Yeah, that was a long-winded answer and maybe pretty vague, but it's the best I can do.
It's pretty amazing to have that space at the ranch to
Fail
live,
at, yeah.
yeah,
Yeah,
to live out these ideas too.
yeah,
Uh,
we can try things, and
yeah.
we can, yeah, no, it's invaluable, and we can, yeah, we have the ranch crew to help out with it, and they're amazing. We can make things happen,
Mm.
and especially now when funding is really hard with universities and things, we're lucky that way.
Mm. Janelle had a question. Have you done any research on restoration without the use of pesticides?
Without the use of herbicides? or herbicides?
They said pesticide.
Mm-hmm. Again, like that would be a better question probably for Chuck, because I'm not really doing, I mean, the only restoration I've been part of is this in the experimental plots. He's the one that is doing the big scale restorations with, I think because the yarrow died, we didn't have to use, herbicides to kill it. But I have not been involved. Like that's the backdrop, right? With all this restoration is that it often requires herbicides,
Mhm.
lots of it. And we've done some studies actually, because I was thinking like everybody's using herbicides, but we never think about what the below ground consequences are. If you go out and you kill knapweed, for example, knapweed is a good host. We know that from mycorrhizal fungi, and it's often replaced by one of those short-lived grasses. If it's not cheatgrass, it's bulbous bluegrass. So we did a study where we just had sections that we sprayed with Tordon. Either I was going to present, but it was too long anyway, so I cut it out. But we sprayed grass-dominated native plots with either water or Tordon, or knapweed with water or Tordon. We did that replicated areas out on the ranch. The idea being that if you spray native grass-dominated communities, we could look at the direct effect of Tordon because it's not killing the plants. And with the knapweed, we knew that it was going to kill the plants, so we would have both direct and indirect effects of Tordon. And what we saw was, yes, Tordon kills knapweed, and it's replaced by bulbous bluegrass, which is like cheatgrass. I mean, it's a short-lived exotic grass, really poor host. And what we saw was like the AMF abundance was reduced by half in 1 year. So that's another thing to think about with herbicides. Like, we know what happens above ground, but think about what happens below ground as well. And the longer that field is laying fallow, the greater chance or risk that other plants come in and the fungi will die if it's just fallow, right? So I'm not, again, a restoration ecologist. And if there are people in the audience that are, they might completely disagree, because I don't really know what I'm talking about when it comes to restoration. But it would seem like that you want to have that area covered as soon as you can with something that occupies that space, that is not an exotic. And be that a bridge species that would do that, right? It would also keep the fungi alive. So, but no, I have not, and I think that's the challenge to do restoration. I'm sure there are people working on it. I have not, in terms of without herbicides. I think that would be really hard because you need to kill the invaders somehow. And I suppose you can pull, but that is also, you don't go out and pull like 500 hectares. But if it's a small area, I'm sure you could do that.
Janelle also had a question. Do you do anything to target the seed bank after removing invasives from plots?
Yeah, yeah. I mean, again, for restoration ecologist, I guess, yes, that, you would want to try to maybe prompt seeding and then spraying, multiple times. Like if you have the ability to water and then spray, or just like wait for a rain and then spray. So it's a little bit contradictory when I'm saying like try to seed right away. Maybe like taking a few rounds of, the seed bank, getting rid of that, I think can make a big difference. We've seen the hard way when we don't do that and we just have cheatgrass everywhere.
Yeah, yeah. Okay, and Janet Sims had a question. Could you speculate about the different dynamic that exists in the regions, for example, Central Europe, where knapweed is native, between the knapweed and AMF? And she says, I wonder what is different in the soil there that keeps the knapweed in balance with the other plants that are present.
Yeah, that's a million-dollar question, isn't it? Yeah, we've been thinking a lot about that because invasion is fundamentally a biogeographical phenomenon, right? Like plants change ranges You know, they come from China or Europe to here. And one, I mean, maybe you know this already, but one interesting fact is that there are more Eurasian plants that come here than vice versa.
Mm.
And one idea is that Europe has a long history of agriculture, so they have a lot more disturbance tolerant plants. So they are particularly good at coming to other places and invading. Not, I mean, we obviously have some that go the other way, you know, like a goldenrod, Canada goldenrod is one, is terrible. I'm Swedish, it's terrible in Sweden, it's terrible in Europe. Ragweed is terrible there. So we send some over there too. Um, one of the most one of the most popular hypotheses in invasion biology is the enemy release hypothesis. And that states that, you know, like associations with plants and their enemies, be that either herbivores or pathogens, they escape them when they come here. So they escape specialist herbivores and pathogens. They might encounter generalists that just eat everything. But if you think about, like, have you seen a deer eating spurge and you know, or knapweed? No, they don't. But in the native range, they do, because they've co-evolved. I guess we can get goats to eat spurge here, and I've seen it sometimes with poor goats. It's this like white sap. But so that is I mean, in terms of mycorrhizae, we didn't think that mycorrhizal fungi played a big role in invasion because the associations are more generalist. So as long as there are mycorrhizal fungi coming, or where they arrive at, and if they're mycorrhizal dependent, they should do fine. We're just now learning in projects we're currently working on that there is a link between escape from herbivores and increased association with mycorrhizal fungi. Because that all that carbon that you use to defend yourself against herbivores in the native range, you go to the non-native range, you escape them, then you have all that carbon that you can then allocate to mycorrhizal fungi. And that's behind that enhanced mutualism hypothesis that I was talking about, that some plants, and it's coupled to enemy release. So we're right now in the process of setting up a collaboration with China, where a lot of these plants are native and then they come here. And then we're going to do that by a geographical comparison in terms of mycorrhizal colonization and herbivory, and trying to link those two. Because it's been speculated upon, but not really investigated much. I think there's only one plant species that they've looked at.
Mm.
Yeah, but it's
I'm
fascinating. It's
glad.
fascinating. I mean, what Alex was saying, like, are you going to give us some hopeful glimpse? And I'm just like, no, I'm just going to present how amazing these plants are. I mean, they do amazing things. So,
Yeah.
you know, think about that. When you see knapweed and spurge, like, they have fundamentally changed who they associate with and how they associate with microbes, and in ways that help them. And yeah, we can learn a lot from that.
Mm.
I don't know if it's going to help us combat them, though. That's the bummer about all this, but it's been fun to kind of dig in to understand what they do.
Well, I mean, like kind of like what you've been speaking to, your interests and your line of work aren't necessarily like seeking a fix. You're seeking information, but that information lays the foundation, right?
Yeah, that's what we
For
write in all the
more,
grants, isn't it? Grant
yeah,
applications.
and then different brains come along and
Yeah.
they think about it and have new ideas. I
Yeah, I think
I've
I think biocontrol is a good example where,
yeah.
you know, studying these herbivore interactions, that has also given us a lot of information about biocontrol. Like,
Mhm.
essentially, we just bring the herbivores back here,
Yeah.
or pathogens. But it takes a lot of background research before you should release a biocontrol, right? Because it requires that it's very, very specific
Yes.
to the invasive plant. Otherwise, it's just going to spread. Like, there's some terrible examples, like the cane toad in Australia, where that biocontrol has just gone away. And they just didn't do the background work that was needed before they released it. So I would never be bold enough to release a biocontrol, but it has been done. I think I don't know if there are 10 different biocontrols on knapweed, or maybe even more. So
Um, if anyone's interested in learning more about biocontrol, if I haven't already told you this, I work at Rocky Mountain Gardens in Missoula, and the Montana Biocontrol project has a couple hoop houses in the gardens, and they are working to um, get a particular little weevil uh, kind of the get it stamp of approval. It's been like a years and years process, and it's approved in Canada already, but
oh yeah, yeah.
for the
What?
rush.
Yeah. I've heard about that.
Yeah,
I've
so
heard
so
about
they're
some people
growing.
wanting to bring like smuggle it across
Yeah.
the
So it's all
yeah.
above board. It's all legal, but they've been growing flowering rush, which has been fun to watch. And
Yeah.
I think they have like 12 mature adult weevils at a
Oh
time. It's like
yeah.
very
Oh
they
wow.
be
Yeah,
very
that's not
careful.
much. Yeah. Huh.
But it's pretty cool, and there's some good interpretive signage out there to kind of share about the program and
Yeah, I
what
have to come
they're
and
doing.
visit. I would love to.
Yeah.
Yeah, maybe I can get a tour
Yeah,
sometime.
you got to
Yeah,
come. I'll give you a tour anytime.
that'd be fun. That'd be
And
fun.
and for anyone who cares to know, we'll be opening our doors to the public on April 26th. Okay, that's my
Oh,
only plug.
yeah,
Um,
good.
we do have one more question in
Okay.
the chat from Lauren. If Ilva, do you have time?
Yeah.
Okay. Lauren says, with a lot of areas in the U.S. having success researching using targeted grazing and prescribed fire to benefit soil ecology and restoration, have you used either at your site or do you plan to?
Ah, that's a good question too. No, I haven't. And I don't know, like with grazing here, because the weeds we're working on, I mean, yeah, I know that Libby McClay used goats to graze a spurge. And I've seen it being used up on the North Hills here in Missoula too. I have never used it. And I guess you would have to use goats or something. Like we can't rely on the deer and elk. We got like 500 elk on the ranch, but they are not going to eat the knapweed and they're not going to eat the spurge. And with fire, I don't know. I don't know. I don't know enough about that literature to talk about it, how successful it would be and with what species, because I could see like the risk with fire is that does it kill the invader? It depends. Like I don't think it would kill spurge. You know, a grassland fire is pretty fast and doesn't go very deep, and they have deep roots. So my fear would be that it would just grow back right away. Maybe with cheatgrass. I mean, I know we've used it a little bit on the ranch. The restoration, they go out and burn some areas just to get rid of the thatch of cheatgrass. I don't know if it's so much to actually kill seeds or something, but get rid of that thatch that forms, and then you can seed into like the soil and get good soil contact. So I know they've used it for that a little bit. You know, fire in this area is also kind of scary. I don't know. It could just take over pretty easily. But yeah, I don't actually know. Like fire also releases nutrients, right? So that would be the other concern if you have like weed propagules there, that they would probably outcompete any native propagules. I know it's used though, but I have not used it and I don't know enough. So I should just stop talking right now.
Well, these are all really these are fun ideas to think about. It's
For
it's
sure.
sparking my thought process about new topics for next season.
Yeah.
Anything can be about native plants.
Yeah. We're doing some fun work with ectomycorrhizal networks and ponderosa pine as well, and
Oh,
looking at carbon allocation, potential trade-offs between carbon allocation to support the network and being able to maintain water potential. Because it takes
mm.
carbon to take up water as well. So there could be a trade-off between allocating to mycorrhizal fungi and allocating to these non-structural carbohydrates to allow you to take up water. So that has been fun. And
Well, I
mycorrhizal
guess you'll have
networks,
to
they're
come
kind
back.
of in the literature now with the mother tree and all that. So
Yeah, yeah. Oh, I don't think you can ever hear too much about that stuff. And it's always nice to have it repeated.
yeah, that's true. That's true. Well, thank you all for coming and sticking around. It's fun. It's always a little weird to just sit and talk to a computer screen, but in a way, it's also less nerve-wracking because you're really just talking to a computer screen.
Well, we were all here and
Yeah,
listening.
yeah, good, 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 good.
All right, I'm gonna stop the recording.